Well, it's an incredible pleasure, and it's all Raghavan who is responsible for this. It's a true honor to have this Raman chair, and it's an incredible amount of fun going around India and meeting students and giving the same lecture. Not quite, but anyway. Um, it's India is for reasons that I do not understand. I feel home. It's dirty, it's complicated, it's bureaucratic, it's everything. But I feel home. It's probably my Indo-European origins that do the job. So it's a true. I really thank you from the bottom of my heart that you have invited me to do this the Raman Chair tour. All right, maybe to check that. So what I will do is I'll give you an overview of Notch, an overview that does not include, much molecular biology and many detailed genetics, even though the genetics and the molecular biology of Notch is in general deliciously complicated. However, I think that this is, especially for a broader audience, this is boring. I don't want to bore you. I don't want you to go to sleep. You might anyway, but at least not because I'll try to do so. So I organize the lecture a little bit into the following. I'll give you some history of how Notch came about, so to speak. I will talk about the developmental logic, even though Vijay brilliantly described it already. We'll talk about a little bit of receptors and ligands, the control of the signal, because that's very, very important, because if anything that goes wrong with Notch, both the development as well as disease is relevant for that. I'll tell you some things, aspects of evolution. Maybe I'm getting old and older by the day, and I must say that evolution is started some time ago to appeal to me, because in many ways evolution will give you the why or address the why and not the how. And then finally, I will tell you some sort of later things about proteomic approaches and so on to try and deconvolute. That's the reason for that. Try to deconvolute some of the complexity of Notch. And during all that, I hope that you will see how complex Notch is. I should also say that if you look at Notch, and instead of Notch, you look at the other fundamental signaling pathways like winglets or hedgehog or whatever, you will see that probably they'll be just as complicated. And so, whatever I say today, anything that has to do with my lab, I did not do. It was always one of these people that deserve the credit. For every accolade to Notch, to me, is because of that. All right. So, Notch. Notch was uncovered through genetics in 1914 by Dexter, who was one of the people working in the magical lab of Morgan, he showed two parameters that I want you to remember. The first one is that there is this notching of the wing, so it's an X-linked chromosome. It's an X-linked gene rather. So females that have one copy, the other one gone, they show this dominant wing notching phenotype. Hence Notch. If we were working on worms, we would have called it UNC 3055 or something, you know. So And the other one is that the males are dead. So this is a lethal mutation. So males that don't have notch are dead. All right. I'm very often asked. Students ask me, not today, but in 3 other occasions, why did I start working with notch? Because for 40 years I've been working on one protein. If you think about it, that's what it boils down to. And the reason is that I have, doing my PhD at the MRC in Cambridge, a magical place in many different ways. Again, we're 10 students and 5 Nobel Prizes. Amazing place. And I was saying that, even if you were a complete idiot like me, just by osmosis, you would get something in your brain in this surrounded by such brilliance. I was finishing off my PhD in protein chemistry, and I started thinking about doing developmental biology, and the means to do developmental biology just had started. Cloning was becoming. Paul Berg, I remember he came and gave a talk at the MRC with R1, doing essentially cloning genes. Very exciting time. And I was thinking of how to do molecular biology in development. So with Jerry, we were talking about that, and he was going to Hogness's lab, who was trying to do that, and I went to Gering's lab. But the reason I got interested in notch is about, I read, while a graduate student, I read a review by Ted Wright, which was the only review in developmental biology. And it said that, if one was asked to choose the single most important genetic variation concerned with the expression of the genome during embryogenesis in Drosophila melanogaster, the answer would have been the notch locus. How can you not be hooked by that? This is like a fishing pole with some juicy bait. And, I was hooked forever, and I'm still hooked in many ways, all right? Since 1914, there was a silence of notch biology until the 30s, when Don Poulsen, that Vijay already mentioned, Don was working on his thesis at Caltech. His advisors, not too shabby, was Sturdivant and Dobzhansky, and he was correlating deletions of chromosomes with phenotypes. And what he found was that doing that, he saw that this is a drawing from Don Poulsen that's hanging in my office. He gave it to me. So he narrowed down notch into notch in the 3C7. Now, a 3C7 over plus will give you the notching phenotype. 3C7, this over homozygous the embryos will die. And if you look at the nervous system with a neural marker, you will see that this is the wild-type nervous system, nicely organized, if you will. And this is a notch embryo, where it's a hypertrophy of the nervous system. But the interesting part was not only it was hypertrophied the nervous system, but it was at the expense of epidermal structures. So a precursor cell in the early embryo that has to make a decision, neural versus epidermis, was making the wrong decision, and instead of having a epidermal and a neural cell, everything became neural. All right. So this switch was severed. All right. Now, given that the cloning had become a possibility, I went to Stanford I went first to Gering and tried to isolate notch by differential hybridization until I realized that this was impossible. Couldn't do it. I went to Stanford. I still wanted to clone Notch. I was distracted by some other things, but towards the end of my stay at Stanford, together with Mark Maskevich, a graduate student at the time, that's Mark. Oops, that's Mark then. Oops, and that's Mark now. What can you do? We started trying to clone overlapping clones and walk from 3C10, which was the protein that Mark was working on, all the way to 3C7. But we were stuck on a wall of repetitive sequences. We just could not do it. To the rescue, now at the time, there was no lab working on Notch. Zero. Nobody. Okay? At the time, except one lab, and that was a cytogeneticist called Bill Welchons, a wonderful guy, and an incredible cytogeneticist, who was studying the region that Notch maps in. And through his work, we were able to get a rearrangement that allowed us to jump over this repetitive sequence. The rest is history. So we cloned the locus. It turned out to be quite big. But before I go on and we talk about the locus itself, I just want to make a few things clear. The first one is that we know already that the embryo is dead. We know about the phenotype, about the hypertrophy of the nervous system, but the wing notching. Keep in mind the wing notching. Wing notching equals loss of function, all right? Because I'll come back to that. We looked at since then, every tissue that has been looked at and developmental event, differentiation, proliferation, apoptosis, and movement is affected and can be affected by Notch signaling. The very central sort of signaling pathway. The other thing one thing that is constant, no matter, in spite of this extraordinary pleiotropy, is that the developmental logic of Notch remains constant, and that is the Greek model. So you have what Notch is doing, no matter where you go, is you have two cells next to each other. One decides to do something, and the consequence of that decision is being dictated to the next door neighbor. So no matter what I tell you, keep that in mind, because that's constant, in spite of this absurd pleiotropy. Of course, that means that ligand cell segregation is essential, which is for morphogenesis extremely important, and it's also important for the definition of boundaries between cells that have different developmental potentials. All right, and indeed, almost all stem cell systems can be influenced by Notch. Almost all of them. There are maybe one exception, as BJ alluded to, but the news are if something does not involve Notch rather than the other way around, which makes it already complicated and to some people boring. All right, so what? I can't do everything. So, how does this Greek developmental, now I keep on saying that now, and Indian developmental logic is manifested? Well, it is because the two cells next to each other express a receptor, a membrane-bound receptor, and a membrane-bound ligand. So that's how the communication happens. And this is a scheme that is absurdly simplified, but it does, it gives you the essentials. So Notch talks to delta or serrate. It's cleaved here in the intracellular domain by presenilin. That was a finding that Iowa Greenwald actually has done. And then the intracellular domain is cleaved from the surface and travels to the nucleus, where it is engaged in activation of or suppression, for that matter, depends, of Notch-dependent gene activity. And again, I want to remind you that abnormal signaling equals abnormal development, and indeed disease. And cancer is one of the pivotal diseases that Notch is involved in, but not the only one. So signal modulation is extremely important. A remarkable thing about evolution is that the morphogenesis, the three-dimensional building of an organism, depends on a very simple, quote-unquote simple, framework of signaling pathways. These are not 1,000 signaling pathways, but there are 6 or 7. And no matter if you are a fly, the president of the Indian Academy, or a fish, you always depend on your morphogenesis, on Notch, on wind, TGF-beta, hedgehog, various RTK forms, JAK-STAT. That's essentially it. This is so the big question is, how is it possible to have the same signaling framework and make a fly and make a human? I mean, control the same morphogenesis. I mean, it is quite extraordinary. And so, because you have, during evolution, you must evolve novel functions using the same signaling framework. A very profound problem, the problem, as far as I'm concerned. Did I say that? Yes, I did say that. It is the problem. Do I have a solution? Absolutely not. Is there any hope for the students that were talking earlier? Is there any hope in solving something like that? And the answer is yes, there is some hope. It might take, not in my lifetime, but there is some hope because of the type of analysis we can do, the type of complicated data that we can gather, and also all the analysis that we can do with this complicated data. I don't want to say artificial intelligence, but I did say it. So this is the kind of things that perhaps this new era of computation might help us do, notwithstanding the difficulties, notwithstanding the. So what I'll proceed in doing is give you examples of control of Notch activity, and it's diverse, but I hope you will see why. So I'll talk first about Deltex and Kurtz. Deltex is a ubiquitin ligase that was fished during molecular screens because it sticks to the intracellular part of Notch. And if you take Deltex and ask the question what it does interact with, you can see that Deltex interacts with Kurtz. Kurtz happens to be a non-visual beta-arrestin. Never mind what they do. Just remember the names, or don't remember them. I mean, so what we know, doing molecular as well as genetic experiments, is that if you have Notch can immunoprecipitate Deltex. I mean, I'll start again. Notch can immunoprecipitate deltex or I'll start again, third time. Notch immunoprecipitates Kurtz only in the presence of deltex. So there's a complex that is being made by Kurtz, Notch, and deltex. If you express Kurtz in the wing, in the margin of the wing, nothing happens. If you express deltex in the margin of the wing, nothing happens. If, however, you express both Notch, deltex, and Kurtz in the wing, you see this notching phenotype, which is loss of function. So I'm destroying Notch signal. Well, what happens in vivo? This is a single layer of cells, happens to be the developing wing, and through the magic of genetics, you can do mosaics. You can do make a patch of cells that don't express Kurtz, and this is the patch of cells that does not express Kurtz. The green is Kurtz in this case. So what happens to Notch? The Notch protein? The Notch protein is upregulated here. You can see it. So the protein where Kurtz is not there, the protein is upregulated. Does it signal? Because that's the crucial thing. All right. And if you look with a signaling marker, here's our patch that doesn't have Notch. Kurtz. Here's a patch also that is upregulated the protein, but only in a specific place the protein signals. Context, another very important concept that I'd like to introduce you to. So if you look now at what Kurtz and deltex are doing, they're kind of modulators. They are not nodes that can upregulate or downregulate the signal depending on the conditions, depending on the environment. So synergy and context modulate Notch. All right, they're not signaled. If you ask now a more sophisticated, a more complicated question, if you take deltex, Kurtz, and Notch and search the genome for genes that will modulate this particular phenotype, and you know that this particular phenotype is wing notching, can I find suppressors and enhancers of niche notching? So this is now a fourth mutation. And the answer is yes. This is the phenotype, that's the experimental phenotype. And we're searching the genome for mutations that will change this phenotype. And sure enough, we can get both enhancers and make the phenotype worse, and suppressors making the phenotype better. How many do we get? More than 100. And if you ask the question, are these all kinases? Are these phosphatases? Are these what? God knows. We cannot make heads and tails out of it. At least not yet. So, that brings me to a fundamental question. How complicated is the genetic circuitry underlying Notch modulation? So how many genes are there that can be mutated or whatever and influence the activity of Notch? So Mark Hankel did these experiments, and he took a loss of function Notch. We are all familiar with loss of function now, the wing notching, and searched the genome. In fact, 55% of the genome of mutations in every single gene, 55% of the genome was mutated in a single way, all right? And combined that with this. and ask the question, suppressors or enhancers? And we know already what everything looks like. And here they are suppressors and they are enhancers. How many of those are? All right? And there are about 500 genes, which is a substantial portion of the genome. Ridiculous. I mean, this is not a trivial answer, all right? But that's what it is. Again, if we try to make heads and tails out of it, nothing, no common elements, all right? So two questions deriving from what I just told you. One is how complex is the overall circuitry of that regulates Notch, and how does Notch integrate its action with other pathways? As far as integration goes, I will give you one example which I always find amusing, if nothing else. So Notch, the screen that we did was with Notch, and two of the genes that we found was E5 and mirror. Again, never mind, they are transcription factors, but never mind what they are. So look at the interactions between these genes and other things. So Notch ARTAVANIS-TSAKONAS is a complex gene that is a known target of Notch, but mirror can also affect ARTAVANIS-TSAKONAS activity. Notch can negatively regulate AROCAN and copolitan. Again, never mind what that is, two genes. But AROCAN and copolitan can positively regulate ARTAVANIS-TSAKONAS. Wingless can inhibit mirror. TGF-beta can stimulate mirror, and positively regulate. And hedgehog is positively regulating mirror. And finally, E5 can positively regulate TGF-beta. So you see that the integration of these pathways are actually quite profound. All right? If you look at, and I want you to remember again that Notch signal integration is bad, is cancer. And Sudhir Krishna, who is an expert in Notch and cancer, can agree or not agree with it. Hopefully agree. Good, thank you, Sudhir. I can't deal with smart questions, so maybe you'll forget it at the end. So, what's happening with Notch and proliferation, right, which is related to cancer one way or the other. So if you look at in a mouse gut, in a mouse intestine, and activate Notch in the crypts of the intestine, you will see that you stimulate proliferation. However, if you inactivate wingless, the proliferation goes away. So Notch has to synergize in this context again with wingless in order to stimulate proliferation, all right? And the same thing is happening in the gut of a mouse, because with a negatively regulator of mouse, APC, that's why we do colonoscopies. So APC over plus will give you adenomas in the mouse, and which eventually will become adenocarcinomas. If you look at the APC mice, develop about 5 lesions per intestine. If we, in this context, we activate Notch, even though activating Notch the mice will be all right, at least for a long time. If we put Notch and APC plus over plus together, then we have 115 lesions. So the chances in developing cancer become much, much higher. But it's not Notch that it is the culprit, if you will, it's the synergy that does the job. And this is a Kaplan-Meier curve. Notch is green, it's fine, activated notch, nothing happens. The APC over plus, it is eventually lethal. And APC over plus with notch there, the mice die fast because they develop adenocarcinomas. An analogous thing is happening in the mammary gland, all right? Analogous. And if you look, so it is notch plus something else that will give you cancer, not notch by itself. I went here. So this is a mouse that expresses activated notch in the mammary gland. Never mind again how it's done. And you develop, we stimulate the development of tumors that are not really tumors. They are benign tumors. And if and pregnancy dependent. If we put the mouse during involution, these respond to involution, and they retract. But if you put it through several pregnancies, at least 2, the mice develop adenocarcinomas and die. If you look cytologically, what we see often is that there's a papillary pattern. This is the benign pattern that eventually becomes adenocarcinoma, the solid pattern on the top. And so again, we see the synergy between notch and something else. We presume that this transition depends on the accumulation of other mutations. We have not shown that though, rigorously, but I think it's the same kind of principle. So how do we look at X? Notch plus activation, notch activation plus X can lead to proliferation and eventually oncogenesis. I should say perhaps that notch itself, part of the complication of the whole story, can be both an oncogene as well as, depending on the circumstances and the conditions, can be not a suppressor of not a oncosuppressor. Well, anyway, suppressor. So how can we find X? Well, X, we can look for X in flies, in the hope that this will define new oncogenic targets. So Palavik Kshetrapal and Diana Ho did a screen. They started with an eye, and I know only Drosophilist can appreciate that this eye is bigger than the control, but trust me, it is. And again, mutate other genes in the genome, combining them with this, and see whether or not we can affect proliferative events. And sure enough, you don't have to be a fly biologist to see that, you can get genes that will push to explode the eye of the fly. In fact, 37 of those genes are metastatic, bona fide metastasis. I mean, and that has been shown and actually published. I mean, at this point. Well, how are they? How many of these things can we see? Well, they found again more than 100 genes that will give you these types of phenotypes. Again, they are not all kinases or proteases or whatever, all over the place. We cannot make heads and tails. But again, so there's no functional commonality. So we were not very happy about that, because in trying to define the underlying molecular causes of these phenotypes, it's one thing to have one gene, or 2 or 3, but hundreds is getting a bit more complicated than we can handle, all right? If you take now, I said in the beginning that there was only one lab working on notch. It was us. It was actually Bill Wilson's, was the only one. Then it was us. Then it was Mike Young, who was also trying to do to clone notch. And that was it. I mean, it was not many people working on that. Of course, now there are hundreds, more than hundreds of people either directly or indirectly working on notch. There is this joke that I've been making that there are two kinds of biologists, those that work on notch and those that don't work on notch. And it's 50-50, you know. So, if you take now everything that has been published, RNAi screens, genetic screens, our screens, your screens, their screens, if you put them all together, we define what we call as the notchome. And the notchome is, so that's all. If you put them all of these together, we see that, what's going on? There are more than 1,500 genes. Unbelievable complexity. And trying to make heads and tails out of that, it's not very easy. Even somebody made out of Greek marble is desperate. I mean, this is, what the hell do you do with that? So there's no one answer to that. But we decided, and I say I decided, against maybe the advice of some of my students, that we should do proteomics. In other words, try to define how proteins interact with each other molecularly and try to correlate that with the genetics that we see. I am aware, so don't ask that at the end, I'm aware that genetic interactions do not mean protein-protein interaction, but it's a start. It's something that we can bootstrap our way up this. So what we decided to do was to define the notch proteome. These are the protagonists. Guru Harsha has been the soul of this project in many ways, and the approach is to take a cDNA clone, tag it with it happens to be a J tag in this case, express it in cells, fish the bait and everything that is linked to it, put it on a mass spec, and define protein-protein interactions. I mean, that's the approach. There's a lot of flaws in this approach, a lot of flaws. This is based on overexpression, which has its own problems. But nevertheless, as I say, it's a start. And it has been a good start for us, at least, all right? So when we did that, we did the proteome, and everything was going okay. What? Until this fellow interfered with the first map. So we went ahead and did a second map. And I will show you the map in a second. And I will go through excruciating detail in every single one of these dots that you see. The colors are groups of proteins that have similar function, or known complexes, because there are quite a few known complexes, known protein complexes. And we have seen we put in about 8,000 individual bait pull downs, and defined, grouped them into complexes. Now these are statistical complexes, are Markov complexes. So any complex that has X number of members, you have to do the biology in order to see whether it makes sense or not. So, and in flies we can actually do functional analysis reasonably well because we can do genetics. So you start with a complex that has one notchome element in it, and you can ask the question, do these other proteins that are associated with this particular complex, do they interact with notch? And you can do that genetically. And I can tell you, sure enough, that the predictability, let's say, of the map is fairly high, in the 50% range, 40% range. Now, what do you do with a map like that, apart from defining new members of complexes if we need more notchome members? All right. There are several things that we are analyzing. First of all, does the map have a predictive value? And I told you already that it does, to some degree at least. And this map doesn't give you solution. This map generates hypotheses, all right? What happens to a complex when individual nodes are disrupted? So you have a complex like that, I disrupt this, what happens to the rest? Does it stay together? What happens? What happens to the network when complex? That's even more important, and it's more significant, because if I interrupt one complex here, if I get rid of this red complex here, what happens to the next door neighbors? Because you can see through these lines, there are interactions that we can identify using this methodology with next door neighbors. So that's the whole system now. So we start looking at the proteome as a system, if you will. All right. So what happens if I disrupt one complex and to the next door connections? What happens to the network when connections and nodes are severed? And what happens to protein quantities? We never talked about quantities. As geneticists, we don't like quantities very much. They are confusing the picture. We like black and white, not something in between. But it's quite important. So the degree to which, but we can again start doing these experiments. We can address these experiments. We can do, isobaric labeling and modulate the quantity of a particular node and see what happens to the complex. Now, I'll give you one, and of course, AlphaFold is giving a different dimension to all of that, no doubt about it. Good or bad, I don't know, but I can tell you that it is an enormous fun to sit down and play with AlphaFold in the complexes and see how everything interacts with everything else. Whether this is correct or incorrect, you have to do the biology, but it's fun. And of course, what happens to conservation? Does a complex, is it conserved in evolution? Yes or no? And of course, before I'll give you one example that we're trying to address. These are two complexes that are connected with one protein. So we're trying to disrupt that and see to what degree, what happens to the connection and the consequences of that, right? But you can start playing these kinds of games. So I'm trying to address the interactions that are defined by that. Conservation, however, all right, that's quite important. So we collaborated with my great colleagues, White Harper and Steve Gigi, And they have been interested in, they hate flies, they don't like flies, and they like humans for reasons that are beyond me, but that's the way it is. And then on the left here is the proteome that we published with Steve and Wade, and the human proteome. Of course, now we can go back to complexes that are defined by either here or there and look for conservation. Conservation usually, not necessarily always, but usually suggests importance, biological importance. So I want to leave you with a totally speculative and probably wrong idea. All right. I mean, is there an integration code? Is there an underlying code like the genetic code, all right, that defines interactions? And the idea is the following. If you look at, is crosstalk, in other words, is it random or does it follow some rules? All right. So here's the idea, and we go back to the Academy of Sciences and its president. So you have a fly, and the fly will define certain interactions, signaling interactions, that will define the framework of cell interactions. Now, in order to evolve, you have to evolve novelty. In order, instead of a wing, you have to evolve, I don't know, liver or whatever, okay? So how is that randomly done? Or is this basic signaling framework still exists? So if you look at you can take this signaling framework and start adding to that new functions, right? And as you go on, you add new functions and new functions. So that at the end of the day, you have fully involved complexity, if you will, all right? But the framework, the main signaling framework stays the same. That's an idea. It's not, don't take it too seriously, or take it too seriously, try and prove it or disprove it. I mean, that's fine. But and this is what we sort of call between us the hyper network, hyper node network. And that is, which for me, again, I'll go back to what I said in the beginning, is the question. Does that really, it's a very profound developmental biology question. That's what it's all about. So, one more time, I thank my postdocs, my graduate students, my friends, my everybody, and I will shut up. I will stop here and ask questions, or rather answer questions. All right? Thank you.
I've been trying very hard to understand other systems of medicine. You know, there are other systems of medicine apart from the allopathic medicine. And in Nature Medicine recently had a paper where they looked at the mind, the immune system, and peripheral lesions working together, you know, and how they regulate each other. And given the fact that in sort of in personalized medicine, you have a lot of variation of the genome independent of a single locus. How do you now think of a process where you integrate a single locus like Notch having different functions with the variant genome in personalized medicine, the complexity of the mind, immune functions, and peripheral lesions working together? I know it's a sort of long-winded question, but sort of fits with your last slide.
I don't know the answer. That's the first answer. But it all depends, I think, on how everything integrates. So if notch interacts with wingless, and this interaction allows me to proliferate, all right, and I go into humans, and it allows me again to proliferate. If I take now, I want a function that does something different. I want this interaction to deal to destroy proteins. Then the constant is the important thing, and I think the context and the way these pathways integrate that depends. I mean, it goes from person to person, or from species to species. I don't have the real answer. I mean, I don't. If I wish I did, I don't. But I think it is remarkable, again, that you have this framework that seems to be constant. It seems to be the same, yet, in spite of that, evolution can do all its tricks. I mean, these pathways are Precambrian. They're not yesterday's pathways. So it seems that multicellularity has that's all it has in its hands to deal with new things.
Yeah. So at a biochemical level, the notch ligand interaction is important, or the strength of interaction is important.
Both.
So how it regulates the multiple networks in both the angles wise.
Well, I mean, the moment you put in quantity in it, which is a reasonable thing to do, things are becoming even more complicated in a sense, all right? On the other hand, we know that the strength of notch interaction, the strength of the signal, can actually give you different developmental outcomes. We know that. There are examples of it.
Wonderful talk, Spiros. Enjoyed it. In this evolution of the hyper network, or whatever information is available from different genomes, your schematic shows just one notch hyper network. Can there not be paraloggers, like notch can be encoded by multiple genes in the mouse or the fish or the humans? And it'll be interesting to look at amongst the paralogs. Can you then decipher a hyper network versus a divergent network?
It's quite possible. But you're making my life even more difficult than it is.
But how many notches are there in the human genome? I mean,
4.
4. Okay.
And that's an often encountered analogy.
Right.
2 duplications of in the mammalian genome. And that's the advantage of life, of course, because you can look at some things naked. But Sudhir's question about personalized medicine includes that, obviously, right? Yeah.
Comes into that.
Yes.
Is there any other tissue type where, just like the wing is just 2 layers of epithelial?
Yes, eventually.
Yeah. So is there any other tissue that one can look at to see whether...
We have systematically, when we're looking at events in the wing, we're always looking them also in the eye, just to look at constancy of the tissue, all right? But we know that a lot of these, even the maps, somehow differ in different tissues. I mean, Steve and Wade have been looking at different cell types and so on. But
So growing up, a student from Vijay's lab, Sumita Anand, and I would often argue how notch signaling works differently in Drosophila versus the higher organisms, the vertebrates. And one such example is that of lateral inhibition. While lateral inhibition is the paradigm for the function of notch in Drosophila,
while lateral inhibition is central to so many things.
So I was going to come to, there may be many more models, a Sri Lankan model, a Bangladeshi model, and somebody else, when it comes to notch in other organisms.
I mean, I can only give you the constant one. I mean, I don't know. I mean,
So
yes,
the so the
it's all
interactome
possible, right?
so the interactome study in Drosophila, would it allow one to now address questions in other organisms,
Yes,
particularly
I
in the
would
field of
say
cancer?
yes. I would say yes. I mean, if you are interested in a particular, well, you see, what we would like to do, I don't know if we will do it, but would like to do, is take now the 10, 15 complexes that are not SOM related, all right, and then bootstrap our way upwards. I mean, disrupt, see how the environment responds genetically as well as molecularly and so on. And again, I want to emphasize that these things are now possible. And you may not need 1,000 people to do it. You may need less. I mean, we have 630 Markov complexes. We can do, quantitative analysis in all of them. It's feasible. I mean, you don't have to kill students for that. I mean, it's feasible. It's not. It's a lot of work, but it's feasible. And I think that biological complexity is something that is inconvenient. It's the inconvenient truth. It's a pain in the neck, and I think I showed you why. I hope. But it's also extremely important because if the control of a notch will give you cancer or not, that's that's pretty significant. So how you regulate notch activity is very important. And if the regulation can happen in so many levels and be that complicated, it's even difficult to imagine drugs. Actually, I mean, there are companies that are trying to inhibit notch as a drug for cancer, and you might be able to do it in specific cases. I don't know, but I must say I would be very reluctant to take a notch drug because it's so pleiotropic. That's all. I mean, it affects every single stem cell system. Now, if I'm desperate, I will do it. I will take it, but or delivered very specifically and so on and so forth. I mean, all these things are possible.
Mhm. Hi. I wanted to, if I here.
Yes.
Yeah, I want to say thank you for your talk. I was wondering if you had to like name the notch system today, knowing what you know, but like give it a different name or maybe translate it into a new language, what will you name it? Because I myself, I feel like in biology class the teachers skipped over the names, and I kind of was sitting there wondering like that seems like a very weird name to have us remember. I was wondering if you had any thoughts about like names in biology.
Thoughts about names in biology. Well, I already made a joke about the worm people, whereas, that everything is Ankh and the number. I mean, in flies, traditionally, and because of Morgan and his crazy bunch of students, many of which got Nobel Prizes, mind you, later, but we were much more colorful. And I think this is fun. To me, I like calling something, I don't know, Kurz, which means short in German,
Yeah.
or something else in Greek or whatever. I think that's fun. But, and fun is important in science.
Yeah.
If we don't have fun in science, science is not worth doing. All right? So, yes.
So I was wondering about this, and I don't know if this is something people have thought about or people have done. So when you're looking at notches along the wings, right? So I could see that in different kind of perturbations, you would have different distance between the notches. So basically, I think that's kind of reflective of the strength of the notch signaling that you're affecting underneath. if that is the case, then you could map the signaling strength from the visible phenotypes that you see back to the networks and look at the kind of perturbations on the network which is giving rise to this different kind of distances between notch. And therefore, you can kind of maybe map the signaling strength from the visible phenotype back to the hyper network that you are thinking about. Is that something? Does that make sense? Is it something people have thought about?
It's not nonsensical. It does make sense that quantity may reflect, reflects all kinds of things, right? But the distance, I mean, the wing notching, um, it's hard to say that this is dependent on strength. I mean, the intellectually dishonest answer is developmental noise from allele to allele and so on. But,
But if you had enough data and enough replicas, wouldn't you be able to distinguish the noise from the signal? So
it could be. It could be. Nobody has done it, so I cannot really answer that in a rigorous way.
Hello, Professor. Really enjoyed your talk.
Thank you.
I was very interested in the point that you made about synergies and how the synergies with notch and other signals are context dependent. So if these synergies are context dependent, how would you then extrapolate the interactors and the outcomes of these signalings across organ systems and across evolution? How would you think about that?
Well, I mean, we know already from experiments done by other people, not necessarily by us, that there are a lot of differences or significant differences between tissues, for sure. Personalized medicine, that Sudhir Krishnan mentioned, again, individual to individual. All of these differences are very, very important and crucial for health and for development. But, in biology, what's important is we try to dissect the most fundamental stuff and bootstrap from that. So if, a particular synergy or a particular integration, signal integration happens, you can take that and go on and dig more and more and more and more and analyze it molecularly, genetically, proteomically, and so on. So these things, again, are feasible now. We can start dreaming of this, of addressing some of this complexity, for sure. And that's why I think that this is a renaissance for biology. I mean, we're saying it earlier, genetics barriers have disappeared. Cell biology, genetics, molecular biology, all the departments here and in Harvard, they're complete nonsense. Everybody's doing everything else. Well, no, but seriously, I mean, there is no, I mean, we hire somebody at Harvard in cell biology, and we compete, if you will, with genetics and with biochemistry. It's all the same, which is very exciting, all right? I mean, this is not it's very exciting. And as we were saying earlier, we cannot be experts on everything. So that's an excuse to be ignorant. But you cannot be expert. So that obliges you to collaborate too. Thank you.
Thank you very, very much, Spiros.
Thank you.
On that note, about the excitement of biology going forward, our ability to observe cells and molecules interacting in action to give rise to phenotypes is something truly transformative now.
Yeah,
So
absolutely.
it's good fun ahead.
Thank you.
Yeah. Thank you very, very much.
So anyway, I would like to, of course, thank Spiros for giving such a wonderful lecture and for agreeing to come to India very readily.
Anytime.
And I would like to thank Vijay, who specially preponed his visit here because he had meetings in Delhi, and he came specially here to be with us today and to
So
be
what
with
will the
Spiros.
Prime Minister do without him?
Yeah.
He said, yeah, that's it.
And thank all of you for coming. And there is refreshments outside where you can talk to Spiros a little bit more if you have questions. So thank you. And we have a little gift for Spiros.
Wow. Raman Spectra.
Spiros is the Raman Professor of the Indian Academy of Sciences. It's, we have just very few professorships, and this is one of them. And it's named in honor of the founder of the Academy. So here you are.
Thank you very, very much. Thank you.
And then there's also a shawl. So.
Oh, wow.
Which
I think I'll need it, actually.
I have to put it around you, Spiros.
Oh, right. Please do.
Yeah.
Thank you very much. Thank you. Thank you.